Okay, we are going to end this week by looking at a relatively new area of study. This is the gut content studies. So this is an area where people have started looking at insects that feed on humans, that feed on animals, and figuring out that we can get some information from what's in their stomachs, or what's in their guts, if you will. So for those of you who are thinking about new and interesting ways to go on and study in entomology or in forensics, this is an up-and-coming area. There's not a lot of people who are looking at it, but it has some potential to do some really interesting things. So the idea behind gut content studies is that these necrophagous insects, or perhaps ectoparasites, they are isolated and analyzed for host DNA. So depending on where the insects were found and the circumstances of the crime, this information can lend us some new and interesting facts about that crime. I mean, think about it for a minute. Look at these basic insects that we have here. Right? So we've got a mosquito here. She is feeding on blood. There's the possibility that if we can catch her before that blood is fully digested, there's still DNA. So we'll be able to tell what person or animal she's been feeding on. And there's been a decent amount of research looking at the types of animals that these mosquitoes are feeding on based on their gut contents. These are maggots. Look at those gut contents there. We can tell, are they feeding on animals, humans, both, neither. The same with lice. So that's the idea that we're looking at here. So investigators are starting to think we can do things like maybe an identification of a victim. We find a bunch of maggots but no body. If they still have active gut contents, let's see who that victim is by checking the DNA. So this is especially important if the victim has been moved. If the maggots found, no body, or there are maggots in two different places, were they feeding on the same body? So the DNA in the gut would then give hard evidence of the body's identity or of alternate feeding sources nearby. So think again about an interesting aspect. Let's say we got a body that has been dumped in a dumpster. There's a bunch of meat in there. And so the possibility is that you have these older maggots going from that meat onto the body. Think about it from the forensic entomologist standpoint. It would be really hard to tell which is the oldest maggot. We don't know if those maggots were actually feeding on the body from birth or from oviposition, or if they moved from something else in that trash. If you can do some gut content studies, you can tell, oh, they were feeding on two different things. So these aren't maggots that can necessarily give us a time of death, time of colonization determination. Okay, so we can go through all of that. We can also connect somebody to a scene. So we have this ability to connect a particular person or an animal to the scene. So even if they're alive, let's say there was a mosquito found at the scene that had this blood content or these gut contents, and their DNA matched somebody who said they were nowhere near that area. So if that mosquito isn't one that can fly 10, 15 miles, now we have definitive proof that they were somewhere within this flying distance of these mosquitoes. So that's the type of thing we are looking at with this gut content study. There are limited studies on this topic. Like I said, the blowflies are the most studied, the maggots, of course. So they're the most forensically or medically important species, just because that's what we're used to. So people have been looking at the maggots, but other species have also been looked at. So we look at California. We have looked at the Sarcophagidae and the Muscidae a little bit, so the flesh flies and the house flies in particular. Now, there's limited information on those two simply because they're much less important at the moment in forensics, so they don't get a lot of attention. So that's a wide open area for research right now. Now, the remainder of the research has been focused on the ectoparasites. So remember, an ectoparasite, something that feeds on a living organism on the outside of that living organism without killing it. In particular, we're looking at the Culicidae, the mosquitoes, and the lice, the Pediculicidae. So that's how we're going to say it today. Now, this is due to their need to feed on blood and the length of time that the blood meal stays in the gut. So again, think about these two, the mosquito feeding directly on blood. We can tell if somebody was at the scene. We can tell what this mosquito was feeding on. There actually have been a lot of studies with mosquitoes and endangered species, or trying to figure out what the main host of these mosquitoes are, or trying to track down maybe not easily found organisms or organisms that are hiding from human observation. You check the mosquitoes, you know that they're out there. That's what we're doing there. Now with the lice, this can go both forensics and just sort of general science. So lice are very, very highly associated with their host. Remember back to our insect systematics lessons. So with these lice then, think about a sexually transmitted disease, the pubic lice. You can tell where those lice came from based on their gut studies. So in a sexual assault situation, if you have a lice transfer from the perpetrator to the victim, you would be able to tie that perpetrator irrevocably to that victim because of the DNA. Maybe you don't have any other hairs or anything from that perpetrator, but you've got these lice and they've got that person's DNA in their gut. Awesome. Same thing with head lice. You can track down where these head lice came from. Same with body lice, all of that sort of stuff. So ectoparasites, wide open for gut content studies. Problem with this is, especially with blood feeders, they can take a while to catch them, and it doesn't take long for them to break down that DNA. So that is something we're working on, or better yet, you guys can start working on that. You're the future. Now, the way that we really analyze this DNA is through STR, or short tandem repeat analysis. So this is a standard for the determination of human nuclear DNA profiles. Those of you who've done any other forensic classes or forensic research, you know STR pretty well. So a short tandem repeat, this is a sequence of anywhere between 2 and 13 nucleotide base pairs. They're repeated hundreds of times in a row on a strand of DNA. Now, everybody has these repeats of DNA sequences. They don't code for anything. So they are just sort of in between these different coding regions. So you can have more or fewer than another person, and it doesn't change the way that your body forms in any way because they don't code for any proteins. So everybody has these repeats and they're all in the same place, but the exact number of times something is repeated, that varies from person to person. Now, when we're talking about DNA analysis, these are the things that we use for what we call a DNA fingerprint. The FBI has chosen 13 specific regions of STR, of short tandem repeats, and those serve as a standard. The chances that any two people other than identical twins having the exact same number of STRs in all 13 regions of DNA is about 1 in a billion. So that's a pretty good way of individualizing a DNA source. So this is what the STR areas are. So it's just basically TPOX and D3S1 358. So we've named them something not great, but these are just regions. And the way that we look at it, so here we're starting, this is the region, 5', 3', think way back to your basic bio for that. But let's say we've got a TCAT. So you see how that repeats over and over and over again? And then here's its complementary DNA strand, AGTA. So these repeat, you just count the number of times it repeats. So this particular person has 6 repeats in this region. Someone else could have 12, someone else could have 2, someone else can have 39, whatever it is. So basically, we are running a fingerprint analysis where we count the number of repeating sequences at this TPOX region, at this D3 region, at the FGA region, et cetera, et cetera, et cetera, and you come up with a list of 13 numbers. How many times you have repeats in those areas. So that's a pretty straightforward and really a quick and dirty explanation of what this STR analysis is. So this is what we use for DNA fingerprinting. We use this exact same technology to identify a species of an animal. So right now, there are databases for cats and dogs and various other animals. There are databases for humans who are in the system. There are databases for any animals where lineage is needed. So purebred animals, cattle, horses, etc. So we're building these databases for all these different organisms. What this means then is you can run a DNA fingerprint on a gut content study or on the contents of the guts in an organism and identify the species. Is this human or not human DNA? If it's human, can we run it through CODIS? Can we see if we know who this human is? Or do you have a suspect? Can we match it to that human? Same exact method. The only problem is you have to get intact DNA from those guts. So we have to think about a few factors. Now, to generate these reproducible DNA fingerprints from insects, you have to have enough intact, high molecular mass DNA. So it has to be isolated from the digestive system of the insect very quickly. So the capacity, the amount of DNA that is present, this is affected by the conditions of the feeding. So the storage of the blood or the tissue in the insect and how much food has been digested. So think about, say, a mosquito. She's feeding on blood. That blood is going to go directly into her midgut. Remember, way back to the beginning of the semester, we talked about that. She's going to immediately start digesting it. So if you get a mosquito, say 24 hours after she's fed, all that DNA is going to be digested. All of that protein is going to be digested. But within 5 to 6 hours after feeding, she's still going to have a good blood meal. That is, if she had a good, huge blood meal. If she only had a little bit of blood, it's going to be gone within 6 hours. So see, it depends on what's going on there. So the conditions of how these organisms are feeding, how that blood or that tissue is stored in the insect. Does it go directly into the midgut? Is it kept in the crop for a while and just stored there until it's needed? What is going on there? And then how much food that organism ingested. So say a mosquito can feed on 3 to 6 microliters at a time, so a little bit. Maggots can feed on a lot more. Lice feed on less. It's just the way that it works. So a lot of research has to go into that sort of thing. Then, once you have these insects, the first necessity in this type of analysis is actually, collecting the insect soon after feeding. In the case of maggots, the older the maggot, the larger the crop. So therefore, more tissue is present. So if you need a good DNA analysis or a good DNA sample, go for those third instar maggots. Remember, you don't want to have the dispersal insects, right? Or the prepupal insects, because they're clearing their digestive system. So they won't have any DNA. So you want to make sure they have a good meal. The larger they are, the bigger their crop is going to be. In blood feeders, you need to make sure that they're collected very, very quickly after feeding to ensure that the blood is still intact in the gut. Now, one way to help preserve the DNA in the insect gut is by quickly killing the insects you're going to analyze. So you don't want them to continue digesting. Instead, you want to kill them quickly and then store them in a way which protects DNA from degradation. So again, remember back to your basic information on DNA. This is why we give you all this info on all the base pairs and the bonding and all that jazz in DNA, so you understand how it degrades and how we can damage it. So DNA degrades in heat and when it is not preserved. If you are going to use insects for gut content studies, you're going to want to freeze kill them. So don't put them in alcohol or anything like that. That can cause some problems with DNA. But so freeze kill them and then store them in very, very high alcohol. We're talking minimum of 80% ethanol. So that way they're already dead. They are not going to be breathing in or anything like that, this alcohol. Then you store them in DNA grade alcohol and then you store them at minus 80 degrees Celsius. That is the standard for storing DNA. So once you've done that, you're protecting the DNA as best you can. Once this insect is then collected, you're going to remove the gut contents relatively quickly to make sure that any residual digestive enzymes are not digesting the product even after death. Because once the enzymes are there, they're going to continue going. They don't need energy or they don't need the insect to be alive to continue this chemical reaction. So for maggots, say, you're going to remove the crop whole. And I'll show you that in a minute. For blood feeders, you're going to need to extract the midgut and then smear the blood on filter paper. That's what you're seeing here. And then store this properly for DNA, for PCR. And then you can take punches of it and start running your DNA analysis. This is an example of crop removal in a maggot. So in this picture, we're looking at Lucilius ericata larvae. And this is how someone goes about dissecting out the crop. You're going to use the tissue from the crop in PCR to amplify the specific sites or STRs in order for you to come up with a fingerprint or a DNA fingerprint. And then you're going to compare these sites to a known, either in a database or in the form of a known sample. So this is actually a series of photos from a project to see if tissue from the crop could be used to identify individual people. What they did was they used medical maggots from 8 different patients. They dissected out the crop. They ran STR, so they ran that DNA fingerprinting, and then compared the results to known tissue samples from the patients. They were able to positively identify absolutely every patient. So here you can see, here's the maggot. You can see the crop in these maggots. You can see that it's full of tissue, and then you dissect out the crop. So this is the method that you usually use when dissection. So this insect's going to be dead. You're going to freeze kill it, put it in ethanol. What you're going to do is going to look something like this. Okay? And you want to dissect it out what we call aseptically. So when you do something aseptically, that means without introducing any new contaminants or DNA or anything to the subject. So you don't want to add your DNA in. You don't want to have foreign DNA. You don't want to lose tissue. You don't want to damage the crop tissue and loose extra enzymes into this gut. You don't want any of that to happen. So you have to be very careful. So you use your best aseptic technique, just like you would with any DNA. You start with your dissection. So you're going to start by removing the posterior segments. So you're basically going to cut off the butt right there. And then you're going to make an incision along the ventral surface. That will open up the area for the crop. You'll peel back that cuticle and you'll remove the crop. So the crop is basically a sac in and of itself. And now this is a nice, beautifully preserved sample of tissue. You're going to flash freeze that, keep it in high-grade ethanol at minus 80 degrees Celsius, and you'll be able to run DNA. So this actually does take practice. This part especially, this ventral incision, cutting through this cuticle without snipping into the crop itself can be really hard. So it's one of those things where you practice a bunch until you get that technique down. And then it's just sort of like peeling a grape, really. You just peel back that crop. All right, so that's it for the gut content studies. You let me know if you have any questions, and you have a wonderful rest of your week.